Hi again. Okay, I want to use this frame from Hank's video to discuss the neuroglia in the brain and elsewhere. In the brain, it's called microglia or neuroglia. There's a bunch of cells types that support nervous tissue, that support the axons and the transmission of information. Now, in Hank's video, he talks about these cells being Schwann cells and the spaces between being nodes of Ranvier, which allows for what is known as saltatory conduction of the signal, or saltatory conduction of depolarization, which means it jumps from node to node, making it very fast. He doesn't mention the difference between these long nerves that exist peripherally and the short nerves inside the brain. These long nerves have these Schwann cells. They're in the peripheral nervous system. They are a fat living cell that insulates this neuron, just like the coating on an electric wire insulates it. In the brain, however, we have different kinds of cells. We have gray matter and white matter. Gray matter is made up of neurons that have no insulating fat cells around them. What they have is astrocytes, like this diagram shows. An astrocyte is basically like the plastic item underneath the hood of your car that holds the spark plug wires apart so that they don't rub against each other. You'll see similar things on high tension lines. That's what an astrocyte does. So I think of an astrocyte like if you stick your arms and legs out like this and you had monkey type feet and you were holding wires apart, that's an astrocyte. Astrocyte, okay? That's in the gray matter of the brain. And Sherlock Holmes talks about, hey, Dr. Watson, it's in the gray matter. I don't know what he's talking about, but he thinks he's associating the gray matter with the ability to reason and think and be smart. In the white matter of the brain, you have fat cells that surround the neuron, the axon of the neurons, continuously. Those cells are called oligodendrocytes. Oligodendrocytes, okay? And of course, the brain is mushy in real life. If you've seen the brains that are made up of in a dissecting lab, if you've seen brains that are preserved in formaldehyde or alcohol, you know they're real tough and rubbery, and that's not the way a real brain really is. Okay, another thing I want to talk about is the geographic or geography of the brain. We know now that different parts of the brain are associated with different functions. And this area that's labeled motor cortex is associated with the ability to move your arms and legs and limbs and other parts of your body. There's another view of this. Okay, so here's the motor cortex. This slice of the brain in here is associated with moving your body. And there are different images that you'll see in the book probably or online that show diagrams of this slice taken out and then across the arc of the brain, pictures of the different parts of the body that are affected here and there and so forth. Same thing with the sensory cortex, which is behind the motor cortex. This is where you get your the sensory nerve endings that sense the smell, taste, sight, and different things. Now, vision actually otherwise is located back in the occipital lobe. This is where your brain integrates what you see with vision. and makes a picture. So if you get hurt back here, you could go blind. The temporal lobe is where you will find speech. So when people have a stroke on the left side, usually the temporal lobe might be damaged, and that's where they have a problem with speech. So what happens when there's brain damage is, in an adult, like muscle cells, brain cells do not regenerate. They do not repair themselves. And one of the challenges for biomedical research today is to figure out how to get nervous tissue to undergo mitosis to repair itself. The best way that your brain handles damage now, if it's not significant or tremendous damage, is that it reroutes the signals around the damaged area. And that's a part of why people with strokes can often relearn to either move their limbs or to speak effectively again. Some can, some can't. Depends on how severe the damage is. But it would be nice to be able to repair spinal cord damage and to repair brain damage so that people could become well again. So one of the main things in our society today is that people who are in certain professions like football and boxing often have early onset Alzheimer's or Parkinson's disease or something like that, or even an inflamed brain, which is actually, we're not sure what causes that, but if an inflamed brain is an autoimmune response and can be treated, but if not diagnosed, would result in death quite often. The other thing I want to talk to you about is something that is not in your book. This is a little extra, and it has to do with Phineas Gage. So if you look at these figures here on YouTube, part 1 of 2, this channel 3 Phineas Gage Didactic, I would like you to watch. You might really enjoy that. It's a reenactment of what happened to Phineas Gage. But the didactic part 2 of 2 is what I think you should watch first, because it really, Alan Alda really introduces the topic here. And so I think it would be better to look at that one first. Phineas Gage basically had brain damage caused by an explosion where a pike used to tamp explosives into a hole went off, the explosion went off and blew it through his head. And he recovered, sort of, kinda, and here's a picture actually showing the path that the pike went underneath the zygomatic bone and out through the middle of his head, and he walked home. But he was hurt. You know, sometimes writings about Phineas Gage say, oh, he just went home and, you know, I had supper and so forth. But, well, you know, it hurt. And he was hurt. But anyway, he did recover, but he never was the same person. His personality was there. And so his personality was altered, and he couldn't function in society anymore because of how the brain was damaged and what part was damaged. And so there's parts of the brain, when we looked at this landscape of the brain again, that have to do with emotion. A lot of that and the control of our emotional responses is in the frontal lobe, and that's where Phineas Gage was damaged. And because he survived this, it became a very famous story. And it initiated what we know today as the as a subject of physiological psychology. I took a course in that at the University of Puget Sound years ago, and I thought it was very, very interesting. But in this field, people are trying to figure out how your brain and what parts of your brain control your thinking and your emotions. And it's pretty interesting. So I thought you would really enjoy watching that. It's a pretty fascinating story, okay? And this involves what we know from biology, and you've probably had a little bit of this in your biology class if you had one, what we call emergent properties. You know, we talk about how life is organized from the molecular level to the cellular level to the tissue and organ systems, and then you finally get an organism. But then there's emergent properties that are our personality, our soul, and concepts like that. And we don't know how that works. It comes from the whole organism, but it can't be identified with any particular structure, anatomical or physiological structure in our body. It's just us. And I've often wondered, if I were cloned and there were 100 of me, would I know us? Would we know me? Would they know me? I don't think so. I think that's a unique thing. Even though we would all have exactly the same DNA, I think we would be different. I think we're a product and part of our environment. And we know now in the field of epigenetics that identical twins are not identical at all. As soon as they're born, they are exposed slightly differently to the environment, and they become individuals on their own, even though they may not look different most of their lives. But they might be different. One might get cancer, the other one might not. And so even identical twins are not truly identical. And so clones of an individual might not be truly identical ultimately. It would be interesting to find out. And I don't know how we would do that with animal experiments. It seems like we'd have to clone human beings. And of course, that's going to be a no-no. But I'm not going to say that somebody won't try it. I'm sure they will. There are crazy scientists all over the world. And it's likely to happen eventually if it hasn't already happened secretly. Oh yeah, there was a book about it. I think Mary Shelley wrote it, didn't she? You remember what that was? Oh, that was a little different kind of a deal, wasn't it? Yeah. Okay. I'm going to talk to you later. Enjoy the week. Enjoy learning about the brain. And if you have anything in particular to discuss, put it in the discussion. Otherwise, ask questions. This can be a really fascinating subject. Okay, thank you.